Thank you, Troy. Our next speaker is Dr. Elly Navajas. Elly is a senior researcher at Uruguay's National Agricultural Research Institute, INIA, specialising in animal breeding and genetics. She has a Ph.D. from the University of Edinburgh. Her research focuses on improving feed efficiency and mitigating methane emissions in cattle and sheep. And since 2023, Elly has coordinated the PROCISUR Working Group on Sustainable Livestock Production with a focus on the Southern Cone. Elly, thank you.
Good morning. Thank you very much for the invitation to be here. Really, it's a great opportunity to talk about the Uruguayan approach in terms of genetics for low emission, and particularly, no, the other way around, yeah. Especially after Jenny's presentation in terms of what is Uruguay, so I just can pick up on what means methane emissions for Uruguay and the relevance of having defined a target specific for beef cattle that implies a reduction of 35% in terms of emission intensity by 2035, and the relevance that genomic selection has been proposed as one of the mitigation strategies to achieve this target. So in this context, what we can redefine in this new context is the fact of the new breeding objectives for our livestock breeding programs in terms of mitigating methane emissions, but without compromising livestock productivity, and also taking into account the relevance of biodiversity conservation and the associated ecosystem services. And why genomic selection is possible? Because in Uruguay, we have had a genetic evaluation system in place for more than 30 years, and for the last 10 years, genomic selection is in place, at least for the main breeds in Uruguay, and I'm talking about Angus, Hereford, Holstein, and Australian Merino, Uruguayan Australian Merino. And the fact that also we have in place phenotyping platforms for feed efficiency and methane emissions. And I'm going to talk about that. So which are the main challenges for us in terms of implementing this genomic selection aligned with the national targets? The first, and it was mentioned before, is the relevant phenotypes. We need to get the relevant phenotypes, even for the reference populations for genomic selection, even if we know that that's the tool that is going to help us improving in both directions, reducing methane and looking after productivity. And methane is one of the targets, but it's not just measuring methane, it's measuring methane in the animals that already have information in terms of productivity traits. We need that information to have the data that we need for the genetic and phenotypic correlations for the selection indices that were mentioned before. And another relevant phenotype is feed intake, that is directly related with methane emissions and is also directly related with animal performance. The approach that we follow here is the fact of building phenotyping platform where we measure methane emissions, feed intake, and feed efficiency in animals that also have information for the other traits, the other relevant traits, and also are animals that are within the genetic evaluation or genetically linked to the genetic evaluations. This is the case of the Hereford Information Nucleus, where you see there Kishu, that is the bull test center where we measure feed intake and run the feed efficiency test. With that information, we build the first reference populations, and since for the last 5 years, we already have genomic EPDs for feed efficiency, and based on that, we started working on organizing 2 experimental herds, which are connected in terms of using the same bulls. Herds that also are associated with different environments. The one that is up, that it means in the north of Uruguay, that is Glencoe, that is on natural grassland, and the one in the south that is on improved pasture, where we collect information in terms of the performance of the breeding herds. And this is a great platform for the next step, that is expanding phenotyping, the methane phenotyping in all locations, to be able to have the reference population for genomic selection, including methane emissions, but also to have the databases that we need for the estimations of the inputs that we need for the selection indices to balance mitigation and productivity. So we started 3 years ago recording methane in Kishu, in the bulls and steers that are measured there, and also collecting rumen samples for the analysis of the microbiome. The big challenge here is to expand the measurements of methane on grazing conditions to also to put together in our database the association between feed intake, methane emissions, and the performance of the breeding herds. And methane, the fact of collecting the rumen sample takes me to the next point, that is the relevance of investigating the association between animal genetics and the microbiome. And that's why we are taking these samples and we are actually running analysis in terms of sequencing of the microbiome to analyze also the role as a proxy for methane. The big point here, and depends on the previous point, is to build large reference initial populations to be able to predict those genomic breeding values and evaluate potential proxies for doing that. Beyond all the platforms and possibilities at national level, it's very difficult for one country to cover all the breeds that are relevant to be able to take into account this effort. And one alternative that is a complementary alternative to the effort, the national efforts, is the regional and international collaboration. In this sense, Uruguay takes part of a Latin American project based on that combines information from Mexico, from Brazil, Embrapa, INIA, Uruguay, and INTA from Argentina, with the objective of building large methane databases of genetically linked populations of 5 different beef cattle breeds. We are also working in the sheep side in another project that is led by Australia. And this is a new project, but also we have experience in the sheep sector, several collaborations, international collaborations, like the one that is mentioned there, that is the Sustain Sheep project. Just finishing, and last but not less important, is the fact of how relevant research funding, public research funding, is for developing these initiatives at international level, but also at national level, and how that is relevant in terms of also linking in communication with the incentive for the private sector, because it's the only way to put together what we call the public-private partnership, when both sides, the public and the private, have the same signal to go in the same direction. And it's important for us to have this. This is part of the different partners in the different projects that we have been involved. And just thank you.